Yeah, so that's a lot of hype, right? Um, but there's this paper called Broad Spectrum Antiviral Therapeutics, which I hope you would agree is a really hyped title, right? So
Hmm.
they made a fusion protein, and there definitely will be a question, but I think the way this should work is I'll just keep talking until you get the idea, and then someone cut me off.
Okay.
Um, so they made a fusion protein which is modular. They have one part double-stranded RNA binding domain. It's human PKR. There's a bunch of these proteins in the cell, and they'll bind double-stranded RNA that's totally not sequence specific. And there's a protein that will activate the apoptotic pathway, which is programmed cell death. And
Hmm.
finally, there's a domain that'll get this protein into cells. So that'll be something like polyarginine. Um, I think there's one from herpes H
Hmm.
something
Yep.
16. They use HIV TAT. I don't know if you know that one. That's an interesting one. But
Yeah.
so
Hmm.
all right. So for the specificity of this drug, the apoptosis is it's going to need to at least dimerize, maybe oligomerize to have activity, right? So the PKRs, you need multiple PKRs, and then that's going to have to dimerize or oligomerize. APAF1 is apoptosis, and then you get the cell death. So that's the key to the specificity, right? So they make a
Hmm.
bunch of these proteins, they express them in E. coli, and they get them into cells. They show they can last for a while, and they do commercial space glow. That's just a bunch of like tissue cultures, right? You're not impressed by that.
Hmm.
But there's two mouse models. There's intranasal and there's
Hmm.
intraperitoneal. And
Yep.
you know, this is high mortality before, low mortality after. They're doing it day -1 to +3 of infection. So that's unrealistic, right? People are gonna present after the incubation period realistically. But we are in a pandemic, so maybe something prophylactic
Hmm.
is good here. But they got groups of maybe 12 to 13 mice. They're getting 60 to 70% survivors with these different Dracos. They make different ones, different segments of protein. So multiple mouse models when you think about it, but they're killing 90% of them with no treatment. And I guess the questions here are: have you heard of HIV TAT specifically as a transduction tag? Are you familiar with other concepts for broad spectrum antivirals with this
Hmm.
kind of range of potential applicability? Can you imagine how viruses might become resistant to drugs like this? And in general, do you think your lab might ever be interested in something like this? And if not, just teach me how to do it and what I need. What do I need? A license? What do I have to do?
Yeah, a great set of questions. So I have heard of HIV TAT, and I think I forget what the herpes protein is, but these are really fascinating because they do have the ability to cross cell membranes and take cargo with them. So in this case, if they can pull in these complexes to drive PKR activation and apoptotic cell death, it addresses one major issue, which is how do you deliver the complex to the cells that it needs to get into. I think because the transition from mouse models into humans is often a long one. You never know if this is going to make it all the way in the clinic, but the fact that they can see in vivo efficacy with it, I think, is very promising. That's a pretty high bar. As you said, as you were describing this, just doing something in a cell culture dish, we can cure an awful lot of viruses in cell culture. And there are a lot of things that will work well in cell culture that as you move toward whole organisms in model systems, they'll fail for a wide variety of reasons. So the fact that this shows efficacy uh, in the animal model is really promising. And you also address the issue of kind of the time frame. The problem with the mouse models is they do have a compressed disease time course in the mouse models. And so you're more limited in terms of just your treatment window. And so I think that that's a limitation of the model rather than a potential limitation of the therapeutic. So, in terms of whether it will go forward, I would want to see it essentially progress through additional model systems. They're hamsters, ferrets, non-human primates, each of which have both their advantages and their limitations. Testing the specificity in terms of just from a safety standpoint to make sure that we're not causing a lot of cell damage or things like that, or things that we would want to look at. In terms of the question of are there other broad spectrum antivirals, so remdesivir, which is came out very early in the outbreak, does have broad coronavirus antiviral activity, and it works against a few other viruses. So that is, I wouldn't say it's truly broad spectrum, but it shows breadth in terms of its efficacy. There's another drug called NHC, which is also known as EID D2801, I think, which is a nucleoside analog that potently inhibits a wide range of viruses, and that's actually in clinical trials right now for SARS-2. I think that results from those trials are probably going to be released sometime this month, because I know a couple of the clinicians at UNC who are actually involved in the trials, and they couldn't tell me anything, but they did say that they're going to break the code in a few weeks, and I think they're excited to see how well those work. I think your question, you asked me if I was interested as well in broad spectrum antivirals, and I guess for full disclosure, I didn't talk about this, but early on in the outbreak, Ralph Barrick, another faculty member at UNC named Nat Morman, and myself sat down before the university shut down and made it so that we couldn't all be in the same room together. And we were talking about what was happening with the outbreak. It was about this time last year. It was clear that the virus was probably going to get away from us. And because Ralph had been involved in the development of both remdesivir and EIDD 2801 through a drug development consortium that Ralph and I are both part of, we kind of knew the time frame that was necessary for drug development. And drug development is normally a 5 to 10 year process. And so what was clear was that as this outbreak was starting to really take off, we weren't ready for it in terms of having drugs ready to go. Remdesivir hadn't really gone through clinical trials except in some emergency use for Ebola patients. EIDD 2801 wasn't ready for prime time. It hadn't been through any safety trials yet. And so we knew that we weren't going to have much ready to respond. And so what we've seen is a lot of efforts to try and identify existing drugs that might be active against SARS-2. There are a couple of those that look like they might have some promise, but the real issue is we're responding during the crisis and trying to come up with an answer. And what we really need to do is have answers ready ahead of time. And so Matt and Ralph and I established or founded a pandemic preparedness consortium that we named the Rapidly Emerging Antiviral Development Initiative, or READDI, which is designed to really identify broad spectrum antiviral drugs that can be pre-positioned so that we're ready for the next outbreak. So I'm very interested in these sort of strategies. And right now, I think anything is on the table in terms of if it shows promise, we should be exploring it, both for SARS-CoV-2, but also to have them in our back pocket for the next time there's an outbreak, whether that's another coronavirus, or if it's a filovirus or an influenza virus or something surprising. Great question.
Yeah, that's amazing. That's the kind of thing I wanted to hear. I'd love to be ready. Speaking of like catchy acronyms, this thing is, because I heard you need a catchy acronym in virology for everything, but it's called double-stranded RNA-activated caspase oligomerizer, or DRACO, because it's a very strict thing. Like, we're talking about low multiplicity of infection in these cell cultures, and they do 15 different viruses. You know, they got rhinovirus, and they've got adenovirus. a couple of those. They have one DNA virus, but this is a 10-year-old study. So my follow-up question would be, do you think it's possible? I heard a rumor that this guy who created this at MIT, Lincoln Labs, old biodefense lab, I guess, that he had let this lapse into the public domain. And I often get accused, just if I try to bring this up with people, they'll say, you sound like you're making a conspiracy theory that the drug companies don't want to invest in this. But on the other hand, I don't know of anybody claiming that you could ever fund phase 3 clinical trials without a patent. So what do you think of the political economy of some of those issues?
Well, so I think that this was part of the discussion with READDI, because you actually get at another important point. If you're a pharmaceutical company, you're essentially, your job is to make money, right? Especially if you're a publicly traded company, you have a obligation to make money. And trying to convince somebody to make drugs for viruses that haven't yet emerged doesn't sound like a money-making proposition, because there's too much risk. The line Nat always uses when we're talking about READDI is, if I had come to you in July of 2019 and told you I wanted to make a drug against SARS-2, you would have said, what's SARS-2? And why should we pay you to make a drug against it? So there's a problem with the entire drug development system, in that there's very little incentive to prepare for these new viruses, because it's hard to prepare for viruses that we don't recognize yet. And so I think strategies like the DRACO approach, well, actually, I'm going to step back for just a second. The other problem is, when a virus emerges, there tends to be an energized response. So because we're responding to the emergency, and so we're now going to see, and we are seeing, tremendous public investment. Again, the vaccines are part of that public taxpayer-funded investment, which we're starting to see the payoff from that now. But then, over time, priorities change and interest wanes. And so it is, I don't know the facts around the Lincoln Labs and the DRACO system, but it may be that funding dried up because it was given less of a priority. And in fact, many of the pharmaceutical companies that we've talked to as we're building out READDI, which we're trying to develop that as a public-private partnership, where it's academic labs, foundations, and pharmaceutical partners, because you really do need all of those players to develop these antivirals and treatments. It may be that the money dried up because a lot of the pharmaceutical companies had disbanded their antiviral development programs because they didn't see it as something that was financially stable for them. And now we're seeing them restarting those programs because, of course, there's a tremendous need with SARS-2. But I worry that the cycle will repeat itself, and that 5 years from now, when we've gotten over the shock of this, we will again see a waning of interest, a waning of funding streams to keep these things going. And that's what we're trying to overcome with this. I think we do have to learn from what we didn't do well in this outbreak. And we need to make sure that we have, that we're positioned to respond much more effectively next time. And our initiative isn't the only one like this. There are at least two other initiatives. One is built around vaccine development, trying to develop broadly protective vaccines, and there's another initiative around broadly effective monoclonal antibodies. So I think these are all complementary approaches. But yeah, I think you don't even need a conspiracy theory to see why some of these things, like that system, hadn't moved further along.